Okay, so do I have to admit? I know someone else is admitting. I saw something come up. Okay, so welcome everyone and thank you to the organisers, Clarice and others, for inviting me to talk to you about quantum genes. That's what I'm hoping to talk to you about today in the next 45 minutes or so. Okay, so genes. I'm just going to move my my screen over so I can see better. There we go. So genes have not as long a history as many other things in science. They were really discovered at the end of the 19th century by Gregor Mendel, famous monk, and when he was working on peas, hereditary at that time was well known. People knew that stuff was inherited. You looked either like your mother or father, but no one had a clue how. I mean, crazy ideas were going around, and because of a good explanation, how people knew by then that, for example, the information passing from one generation to the next was either passed in an egg cell or a sperm cell, but they were tiny. And how do you get all the information to make a person into something so tiny as a sperm cell or an egg cell? That was a real mystery. And people played around with lots of ideas, including tiny people inside the egg that hatch out. So there was lots of crazy ideas around. But anyway, Gregor Mendel was studying the inheritance of peas in his monastery, and he noticed a strange feature about them, that really the inheritance patterns, whether a pea was round or wrinkly, etc., and lots of other features like the colours of the flowers of the peas, they came in whole number ratios when they were inherited. And that was a bit odd, because stuff in biology tends to be continuous rather than digital. Range of colours of people's hair or heights, and not these whole number ratios. So if you crossed a round with a wrinkled pea, he would get a 3-to-1 ratio. When he mixed those, the individuals of that generation, then he got a 1-to-2 ratio. So these ratios changed from generation to generation, but always these whole number ratios, which is rather strange. Anyway, at that time, of course, he didn't have a clue how genes were inherited, but he did discover the gene, essentially, at least in an abstract kind of way, as being whatever was causing this whole number inheritance. Right. Okay, that's interesting. My cursor isn't going forward. Why done that? Yeah, a second. I'm just going to go out of presentation mode for a moment and see if that cures it. Okay, let's go back in again. Here we go. But mutation, we know, and I've known since, that mutation is an absolutely vital part of life. It's responsible for evolution. Without mutation, we wouldn't have evolution. And so over 3 or 4 billion years, the simplest organisms have developed into complex organisms like us and horses, and entirely through mutation, kind of random walk in the space of possibilities of living organisms. So mutation is involved in evolution. It's crucial for evolution. And that has generated the vast variety of life. This is Carl Booth's 3 kingdoms of life. And down here is the ancestor of all life, Yucca. It's called last universal common ancestor. It has given rise to all of these different kingdoms entirely through mutations. And here's our animal kingdom. Here, a very small twig at the end of this very deep tree that goes back billions of years. So mutation is responsible for biodiversity. It's also responsible for disease, such as cancer, as we know, And we have somatic mutations, mutations that occur in our somatic tissue, which is our colon or liver, etc. And those mutations cause cells to grow in an uncontrolled manner to cause cancer. Also, genetic diseases are caused by mutations. And again, they show this whole number of heredity that Mendel discovered. But at the time that Mendel discovered it, and for a long time afterwards, still no one had a clue what genes really were. That all changed in 1953 when Watson and Crick discovered the structure of DNA. It had been known for a couple of decades that DNA was the hereditary material. Experiments by Avery, for example, demonstrated that DNA could transfer characters in bacteria from one organism to another. But that entailed a real problem then. How does a chemical that you could isolate in the laboratory and it comes out as a kind of white powdery, papery kind of material when you dry it, how does that encode everything that you are in terms of how you were born, at least? How does a chemical do that? And that was a real problem for the early decades of the 20th century. And it was solved to a large extent by Watson and Crick's structure of DNA that became the fundamental building block of molecular biology. And here's a genetic letter, of course. The first question we asked is how is genetic information encoded? And the structure of DNA provided the answer in complementary bases. And a typical DNA-based pair with adenine on one side and thymine on the other. And they're held together by hydrogen bonds that, if we go back to the structure of DNA, the chain is formed by phosphodiester groups, and then the letters of DNA formed, the chain really holds the letters of DNA in place. But the key thing I want to point out at this stage is what the letters really are. How is the genetic information actually written in DNA? And basically, it's the position of these hydrogen bonds. Hydrogen bonding is the basis of genetic coding. It codes for how one base will interact with another base in heredity, and it also encodes how a base will give rise to protein information. So that is all based on the position of these protons in the DNA bases. And of course, protons are mechanical entities. So there's already a flavor of quantum mechanics, at least in DNA. And that's also, by the way, also should point out the fact that DNA came in two strands and separates, as we know, and comes back together, of course, solved the problem of heredity that Mendel had discovered. So, as I was saying, DNA is really, the genetic code is really a proton position code. And you can kind of think of it, if you remove the backbone, then you just have the proton positions. And that is reading those proton positions in relation to the backbone, you could read genetic information. So it's really genetic information written in the position of protons. And it's a very old form of genetic information. It goes back nearly 4 billion years. So when, for example, artificial quantum coding was invented in the late 20th century, IBM famously writing their letter in atoms, living organisms have already been writing information in terms of position of fundamental particles, protons, for billions of years. So it's an ancient form of quantum coding, present in all living organisms. Now, back to the mutation. So once DNA was discovered, then mechanisms for causing mutation were also became apparent. Lots of different agents were already known to cause mutations, such as oxidative agents, ionizing radiation, mutagens. And these were all found to interact DNA. And these are what we call physical mutagenesis, caused by physical insults, if you like, oxidative damage, ionizing radiation, mutagens, heat also. And the key feature of these mechanisms of mutation is that they're random. If you think about, say, oxidative damage to DNA, it's going to occur entirely randomly along the DNA strands. And all of the other mutagens, such as heat or mutagens, are just incorporated randomly into the DNA strands. So this is important because the fundamental tenet of neo-evolutionary theory is that it's really made up of two processes. So we talked about evolution and the importance of evolution in biology, of course, but there's really two processes that make it work. One is random mutagenesis. So that generates variation, and then natural selection kicks in to select the fittest variants. So that is the fundamental tenet of molecular biology, and there's also the fundamental tenet of evolutionary theory, that mutation is random and the direction of evolution is provided by natural selection. So it's a random walk plus selection. That's really the fundamental dogma of how mutations occur. And as well as mutations occurring due to... Sorry, did someone say something? If you did, I didn't hear it. Okay, so as well as the random effects of agents, physical agents such as radiation, even in the absence of all of those, mutations occur because of DNA replication errors. And in organisms such as us, and even bacteria, simple bacteria, that error rate is actually very low, about 10 to the minus 8. It's a really low, extraordinarily low error rate. But it does come up with mutations now and again. But those are, again, as far as we know and as far as we understand, they're also random. And but what causes them? So we know what causes the physical mutations, the physical mutations like radiation. We know what the agents of mutations are, but why do random mutations that occur during DNA replication? What causes them? It was a question that Watson and Crick asked. And what they came up with is this hypothesis, one of their famous papers: mutations could occur because of a base occurring very occasionally in one of the less likely tautomerik forms at the moment when the complementary chain is being formed. Okay, so that was in one of their 1953 papers. And this is a base pair between adenine and thymine, and that's a correct base pair. That's a base pair that isn't a mutation. That's every adenine should have a thymine opposite it and vice versa. But if one of the bases go into a tautomeric form, and for example, you can see here in which one of the protons attached to this nitrogen moves over to here, then adenine can pair with cytosine. And what Watson Crick suggested, if the base happens to be in the tautomeric form when DNA replication takes place, then it can generate a Watson and Crick base pair configuration, but a wrong one in the sense that it's not the right base pairing in DNA. Adenine should pair with thymine, cytosine should pair with guanine, and here is a mistake, and that could be caused by tautomerization, one of the bases being in its tautomeric form. And the tautomeric form of bases was known from basic chemistry for many decades beforehand, but bases and many other chemicals can spontaneously form tautomeric forms. And This is how they're normally drawn in chemistry, that, for example, aniline can go from one form to another by this proton moving from here down to here, no, over to here. This proton moving from here over to here, or in here, the finally can go into the enol form. And what is going on here in these tautomeric forms? I must admit, when I was taught chemistry, we were kind of told, well, the tautomeric forms are structures of molecules that spontaneously switch between one form and another. And that's why you get these double arrows. But is that really what they do? And this is what we're going to be exploring. And here on the other side is tautomeric forms of cytosine, adenine, etc. So each of the bases can form a tautomer, can be found in a tautomeric configuration. And in that tautomeric configuration, it may pair with the wrong base. So that's all fine. And that became a standard model for how spontaneous mutations may occur. The base, during DNA replication, the base happens to be in the tautomeric form, either in the template or in the incoming base, and that can cause mutations. But it was only a theoretical model. There wasn't really any evidence for it. And I became interested in this way back in the late 1980s with this interesting paper that was published by Johnjoe. He's a very eminent bacterial geneticist, well, geneticist generally, molecular geneticist, worked on both viruses and bacteria. Worked at Caltech, not so far from you guys at UCLA, and many other eminent institutes like Harvard and Oxford. And he did something that generated he did a simple experiment, but it generated a rather odd result. He grew a Lac minus strain of E. coli. So Lac stands for lactose. And normally E. coli can eat lactose and grow on lactose. But this is a mutant of E. coli. It can't grow on lactose. The fact that it's a mutant isn't important in that sense. It's just unable to grow on lactose. However, if he plated these bacteria onto plates containing lactose and left them for quite a long time, then what he found was he would obtain colonies of lactose positive strains, mutants of the lactose minus. It's got an inactive lactose gene. But if he left them on plates with lactose, then the mutants appeared. Now, that in itself isn't at all surprising. That's what we expect from evolution. What was odd in his experiments was he also plated his E. coli strains on media without lactose. And in that condition, they didn't generate mutants. That's a more difficult experiment to do. He had to constantly take the E. coli off and put them onto media to see, are there any lactose, any mutants there that can grow on lactose? And what he found was no, it stayed pretty much zero all the time. But in the presence of lactose, these lactose positive colonies kept popping up. And this was extraordinary. And everyone was in microbial geneticists like me at the time was completely baffled by it. So in Kearns' paper, in Nature, we briefly review the source of this idea and then describe some experiments suggesting that cells may have mechanisms for choosing which mutations will occur. And this is biological heresy. What he was talking at the time really was Lamarckian inheritance. This is the idea going back to Claude Lamarck, the famous French biologist, the first person to call biology, who was a very great biologist, but he did come up with the idea that organisms evolve by kind of wanting to evolve in a certain direction. Famously, he imagined an antelope that stretched its neck to eat the leaves of trees, And that was inherited by its baby antelopes, and they stretched the neck a bit further and a bit further until they became giraffes. So this was called Lamarckian inheritance, and it was lambasted in the 19th century and discredited. And then in 1998, John Cairns was finding a giraffe on his E. coli plates, in that it was it seemed to suggest that E. coli could kind of choose which mutations it would most benefit from. So instead of having the rate of lactose mutation random in respect to the environment, it seemed to only happen when lactose was present and the cells could grow. And this was, as I said, biological heresy, a furor in the biological community, and everyone was stumped. Anyway, I was a microbial geneticist at the time, so I became interested in this. And I was reading, strange enough, John Gribbin's Schrodinger's cat, and finding out about quantum mechanics. So I was wondering if a quantum mechanical, this is...
May I ask a question? I'm sorry to interrupt.
Sure.
So for the mutations to arise, the cells have to be growing to some extent, right? Because they have to be replicating their DNA. So is there something in the plate that allows them to grow to a little bit?
Yeah, the cells don't die. They go into a state of, we would call it kind of dormancy, in which the cells go and they tick over metabolically very slowly. And they can remain on this state for many weeks, even years. Some bacteria will remain alive with a very low metabolic rate. And quite what's going on there in these cells is still being worked out. But it's also known that some cells are dying and they're releasing nutrients, other cells are still replicating at a low rate with the nutrients that are being released. But you've hit a nail on the head of a really important point here. What are these cells doing in this state? And this remains a bugbear of the whole experiment. But
thank you for clarifying.
Yeah, but this is another way in which the heresy of this idea was apparent. This is what's called the central dogma of molecular biology, that information is stored in DNA. It's transcribed into RNA, the kind of mirror image of DNA, that takes the message to the ribosomes, where the information is used to make proteins in translation. So that's transcription and translation. So the information is only in this direction. And this was the central dogma of molecular biology. In fact, it's had to loosen its dogma status more recently, but at this time, there was just no way that anyone could think of that would allow the environment to change the DNA. There was no feedback to the DNA as far as we knew. So it was really difficult to see how the presence of lactose in the media could influence what was happening in DNA. So this was a big puzzle, and lots of people pondered about it, including me. But I did know of the work of Pierre Olaf Lodin, and he was the first person to really point out that DNA genetic information is actually written in proton position, and there is a kind of quantum code. And and it actually reflects an earlier idea that Erwin Schrödinger, and we'll go into his work, but he had proposed a decade or so before the structure of DNA was revealed, that heredity should be based on some kind of quantum code, what he called a aperiodic crystal, in which single quantum entities such as atoms would play a role. Atoms or fundamental particles would play a role in heredity. And that was, of course, borne out by Watson-Crick's structure. But he also proposed mutations may occur by a quantum mechanical mechanism. And Lodin took this up and proposed a mechanism by which DNA protons may tunnel to make quantum tunnel rather than a classical route to the tautomeric position. And this shows the kind of thing that he imagined. So it's a double proton transfer. You need a double proton transfer to maintain the charge. across the double helix. The set maintains the same charge. So two protons move, but in opposite directions. So he proposed this double proton transport mechanism, but as in the last point here, many theoretical studies have supported this as a plausible mechanism, although not all, but it's not really been shown to operate in nature. Now we're going to go on to look at it a little more detail. So what we're talking about is a proton motion, of course, and like all quantum mechanical motions, it's fundamentally quantum mechanical. It's a motion of a particle, but the real question here becomes whether this proceeds by a quantum mechanical tunneling through a barrier. So we're talking about a proton being on one side of the barrier and having to go on the other side of the barrier as it moves from here to here. And it can do this by tunneling, but it can also do this, and this is what most chemists have been thinking about in terms of understanding tautomerization, that it may go over the barrier. If there's enough heat energy in the system, then maybe the proton can be just pushed over by non-quantum mechanical mechanisms. Of course, everything's quantum mechanical ultimately, but by classically understood mechanisms, simple motion. Okay, so that's the question, whether the transition from a normal to a tautomeric state to cause a mutation may proceed by a quantum mechanical mechanism, tunneling, or simple classical mechanisms.
Johnjoe,
yeah.
I have a question. In theory, is the rate from the C to G the same as the one from A to T?
No, not necessarily. I will come to that in some of the theoretical work that we'll be looking at. So most people thought not, and that's because most people thought that this transition, if it happens in DNA, that it's likely to be caused by classical mechanisms, and that's because tunneling, of course, it's a quantum mechanical process, and depends on coherence, and coherence, as we know, is a very delicate phenomenon. It can be easily knocked off kilter by, for example, simple heat. And living systems are, from a physics perspective, hot. This shows a cell membrane and a simulation of the motion in the cell membrane. And as you can see, it's a very busy system. And all of the cell is kind of in this state of constant motion due to the Brownian motion of molecules. And in that kind of system, we wouldn't expect coherence to last long enough, or at least as a first guess, to last long enough to allow a coherent proton to travel from one place to another. Okay, so with that idea, this is putting your head in a lion's mouth. So I came up with this idea that quantum mechanics may be involved in this process of adaptive mutations, and an idea for it. And we'll see what the idea was in a moment, and then we'll look at a later work. But I thought I'd find out about, I thought I'd discuss it with physicists. So at the University of Surrey, where I work, I got in touch with the physics department, and from a biologist perspective, my head in the lion's mouth by giving a seminar on what I thought might be going on, which involved quantum tunneling. And Jim Al-Khalili was in the audience. So, first of all, the audience was very skeptical, but at least polite. But Jim was in the audience, and he became sufficiently interested that we then met up every now and again to look at this mechanism, which I was thinking of. And it's really one which was very back of the envelope. And this is what it is. The idea was that in media which there was no phenotypic difference whether a mutation had occurred or not. So for our starting lac minus strain of E. coli, if you put it on media that doesn't have lactose, it doesn't care. So there's no measurement of whether the DNA is in the non-mutated form or the mutated form. So in the absence, there's no phenotypic difference to the non-mutated or mutated. And therefore, there's no quantum measurement, was the idea. But when you add lactose, suddenly the system can measure the state of the DNA. And what Jim and I proposed was that this measurement would cause increased coherence of the system and kind of drag the system over into its mutation state. And it depended, of course, on the system being coherent in the first place, that it could transfer over easily over from one state to another as a coherent mechanism. So, but it clearly was a very woolly, back of the envelope kind of idea.
I have a question. What do you mean by coherence here? I'm not really sure I understand what sort of coherence you're referring to. Coherence in what?
The coherence in that, if we thought of the proton transfer going from the non-tautomeric to the tautomeric state, as I understand it, coherence then is saying that both states are present at the same time. The wave function of the state includes both states, with some frequency between them. Does that make sense? So it's essentially, we're talking, if you go back to the position of the proton, for example, in this, the proton is not here or there, it's actually here and there at the same time. It's a coherent state.
Some sort of superposition of having
a superposition of the proton being in one place or another in the DNA strand.
And your assumption is that there are some mutations that can keep this superposition of back and forth tunneling going for longer?
Essentially, it was, I think we shouldn't spend too much time on this because we've kind of really abandoned it, but just showing where the idea came from or how we got into this. The idea was that in the absence of lactose, the cell can't measure whether its proton is here or there in a quantum superposition, so it's unmeasured. So it can constantly swap between the two states, if you can think of that, but it's really present as a superposition of both states. With lactose present, then you've got a measurement. And that was kind of forcing the system to choose one way or another. And we had some very back of the envelope idea about how that might increase the number of mutants. But let's not dwell on that because we don't really go with that idea anymore. But it got us to working on DNA. So we published this paper, and I was inspired enough to write a book about quantum evolution based on it, rationally. But life gets complicated. Life is always complicated, and living systems are the most complicated entities that we have. So even a process like DNA replication is very complex. For example, when we talk about a base substitution happening in a DNA molecule, there is at least 3 different processes that must take place. First of all, the DNA has to be incorporated, the incorrect base has to be incorporated into the DNA. But DNA polymerases also have proofreading. So they have an error correction machinery, which removes ill-matched bases. So that's going on as well. And then there's another process, which is called base excision pair. And that, when a DNA base has been incorporated into the DNA, there are many mechanisms which will remove it. So there's a lot of different things that can go on. And this is how, if you get a DNA mismatch incorporated into the base, into a DNA base pair, then you have this mechanism of enzymes which are designed to remove them. So there's at least 3 different processes that this must go through in order to become a mutation. And any of these could be causing a mutation if there's a fault in any of these. So this is just to point out that it's a lot more complicated than our initial kind of model. And of course, I can't see the title, but okay. But nevertheless, there was at the core of it the idea of the proton tunneling. in DNA. And we thought, okay, that's something that is worth pursuing, whether or not it's involved in adaptive mutations. And adaptive mutations also became rather messy as well. I won't have the time to go into that. But then we became interested in a fundamental question about whether DNA, protons in DNA are delocalized. Are they present in different positions at the same time as a coherent superposition? So, why is that stopped again? Okay. Okay. So, as is shown here, there have been many theoretical studies, quantum mechanical studies, using Schrodinger's various versions of Schrödinger equation to ask this question. And rather than giving you a whole list of them, this is a review that Johnjoe Kim, one of our lecturers in quantum biology here at the University of Surrey, and who's spoken at UCLA meetings, wrote in collaboration with our students, an update which includes a review about whether tunneling is involved in DNA mutation, and it has all the references. There's lots of them, lots of different studies, and many of them agree, many of them don't agree. So the jury from a theoretical perspective seems to be out on this question, but I'll go on and look at the work of one of our students in a moment. Experimentally, you can ask the question, does DNA exist in this form? And there have been, of course, the problem with tautomeric forms is they're present in very low amounts. So it's estimated the tautomeric form of DNA is probably only present in 1 out of 1,000 DNA bases from theoretical studies. So that would be very hard to detect. So it's hard to experimentally prove that the tautomeric forms are present. But some studies have gone on to look, for example, in this study here, that a GC base pair mismatch, a synthetic one, could be incorporated into DNA polymerase and be present in a Watson and Crick configuration in the active sites, showing that if DNA can get into this tautomeric form, it will form a Watson and Crick base pair with the wrong base. So that's an important finding. There's also more recent NMR studies in which they also detected tautomeric forms of DNA. And they also looked at this from the method that I'm sure you're familiar with in quantum biology. One of the ways of probing quantum biology in enzymatic systems, at least, has been to throw, and particularly to probe the question of tunneling and proton tunneling, is to replace the hydrogen nucleus with deuterons, and to do experiments in deuterated water, and or with deuterated synthetic chemicals. And because deuteron has twice the mass of the hydrogen nucleus, a single proton, that usually decreases the frequency of the transition between, usually decreases proton tunneling. Proton becomes more massive, it's much less likely to tunnel. So, so they demonstrated that they could detect the tautomeric form in DNA by NMR, and they also showed that the amount of the tautomeric form was sensitive to replacement of hydrogen, doing the experiment with water or doing the experiment in D2O. They got a different frequency of what they're detecting as the canonical form of DNA compared to the tautomeric form. So it did seem to be that changing the base from hydrogen to deuterium was decreasing the rate, decreasing the transition between the tautomeric forms. And that could be explained by a quantum mechanical mechanism if tunneling is involved, because tunneling, because it makes the base more, the hydrogen nucleus more massive. Okay, so that's a work by other scientists in the field, but in 2018 we obtained funding for forming the Human Quantum Biology Doctor Training Center at the University of Surrey. So as Jim and I have an interest in this, we funded some PhD students to work on this question. And here's one of them, Louis Slocum, who is a theoretician, and he has done some work on this question. And then we're going to have a look at some experimental work. So what Louis did was use density function theory to map the how nuclei interact with the delocalized cloud of electrons forming the bonds. However, I'm getting all this from Louis. This is not really stuff I understand, but I can read the graphs. He then used open quantum system modeling to look at where the protons actually were, because in density function theory, they were treated as a point in space, whereas using open quantum systems, it could map out where the protons of DNA, the coding protons of DNA, were, and also look at how they switch from the canonical form to the tautomeric form. And this shows the temperature transition between the classical to canonical to the tautomeric form at different temperatures. And of course, classical over the barrier transition is very sensitive to temperature. So it doesn't really become very significant until temperatures are very high. But quantum mechanical tunneling, of course, is much less sensitive to temperature. So at normal room temperature, what Louis found was that really the only way, or at least the quantum tunneling mechanism, was 4 orders of magnitude more likely to be responsible for the tautomeric state than any classical mechanism. And so, in other words, if tautomers are forming, according to Louis' calculations, they're forming through quantum tunneling. Now, Louis also, so that work was published in 2021, but further work he's done has looked at whether this will change if we replace hydrogen with deuterons in the DNA in the DNA. And what he calculated by performing his calculations both with hydrogen nuclei and with deuterons in the DNA base pairs, and then calculate their kinetic isotope effect. Was there a difference between the amount of DNA in the tautomeric form compared to the amount of DNA, the amount of the base pair in the tautomeric form compared to the amount of the DNA of the base pair in the canonical form? And what he found was a strong kinetic isotope effect at biological temperatures, that it would be a factor of about 10 to the 3 in terms of the ratio of the tautomeric form to the canonical form. He get kinetic isotope effect of about 10 to the 3. In other words, 10 to the 3 more of one rather than the other, and that was entirely due to tunneling. So that suggested that both that if tautomers are present, then they are generated by tunneling, and also that we should be able to detect the kinetic isotope effect. But he did have some qualifications. So could tautomers survive this process? This is one of Louis' slides, so this is kind of his thoughts about this. The tautomer is continually destroyed and created over much quicker timescales than the biological process, very 10 to the 3-fold quicker. And the quantum reaction rates are large, so the proton is effectively delocalized along the hydrogen bonds. We already know that a hydrogen bond itself is, of course, a quantum mechanical entity. The proton is delocalized across the hydrogen bond. But what he's saying, it's also delocalized along the hydrogen bonds. And okay, and those published. Now, the other very brilliant, Louis was a very brilliant theoretical physicist, and Antonio is who you also know at UCLA and is giving talks in the UCLA seminar series, is an experimentalist. So he came in doing looking at this experimentally, and first of all looking for the kinetic isotope effect. And first we asked a very simple question: if we grow E. coli in ordinary water or deuterated water, would it make a difference to the number of mutants? And this is E. coli and the system, genetic system. Use these purplish dots are mutants, and you just count simple, very simple experiment to do grow E. coli in ordinary water, in deuterated water, and count the dots and see if there's any difference. So the idea again being that deuterium will at least freeze the transition between these two forms. And what in this experiment with live E. coli cells, what Antonio got was there was a difference, that if we did the experiment in water, it got a mutation rate of just below 10 to the minus 6, but then in 100% D2O, it was 2 times 10 to the minus 5 times 10 to the minus 6 compared to 2 times 10 to the minus 6. So it's about half the mutation rate. But biology is messy, and there's so many things that can go on in this experiment that we don't really have control of. And it's certainly not enough of a result to know that really what's going on is anything happening at the level of proton tunneling. There are lots of other processes going on in living cells that may be sensitive to changing the isotope from hydrogen to deuterium, as we know, because when we first grow E. coli in deuterium, it doesn't like it at all, it dies. So clearly it's having a big effect elsewhere in the cells. So you couldn't really conclude anything from that, except it was something interesting in the right direction. So Louis has then set up a more simpler system, in the sense that it's a simpler in vitro system. So what he did was just look at a single reaction in which a mutation is going to be registered. So here we have a base, and if a base goes into a wobble position, we won't go into that, then it can G can be matched up with a T. And what we're looking at is whether that will form a stable configuration, and in water or D2O. So the experiment is set up, and this is yes, okay, is that we take a piece of DNA with a template. This is the template strand, and it's got a G here. And that's waiting for a C to come in. But what we're supplying it now is either a C or a T. So in one experiment, we supply it with a C. In the other experiment, we supply it with a T. And then we do those in both ordinary water and heavy water, deuterated water. And we look to see if the deuteration of the water makes any difference to the level of incorporation of the T. So incorporating the C will be non-mutation, that C's are meant to come in here opposite the G. If T gets incorporated, that's mutagenic. That's the wrong base coming in. So we're looking to see if that is influenced by deuteration. And we're doing it in a very simple in vitro system, so cutting out a lot of the complexity in the previous system. Now, the first thing we had to do, or Antonio had to do, was set it up so as we got the same level of incorporation. And you can see by the concentrations here, this is a concentration curve, and looking at the incorporation of the CTP against the template of a G, and you can get that very efficiently incorporated at very low concentrations of the D to CTP. In order to make this work with TTP, you have to put much higher concentrations in. So we have to titrate this so we got the same level of incorporation of TTP into the template, into the growing strand, as CTP in the growing strand in the non-mutating situation. So in other words, we have to use a lot more TTP in order to force the reaction to incorporate a mutant base, if you like. And then use gel electrophoresis to separate the products. And basically, then this, we go back to this. This is a very short piece of DNA, only 10 bases or so. So it goes to 10 or 11 bases. So the question is, with this this strand that we're making, it's 10 bases long. So if the DNA is correctly incorporated, it will be 11 bases long. And if it's not, it will be only 10. So what we're looking at is the size of DNA that is made and whether that's affected by deuteration of the medium. So you can separate on a high percentage of acrylamide gels a base that's a DNA strand that only differs in one base pair. And this is the kind of result that you get when you scan the gels. And this shows the 10 base pair and then the 11 base pair form. So we tried to get them so about half the DNA goes into the elongated form. No, the cursor isn't working again. Do the same trick, hopefully. Okay, and this is the kinetics of the reaction. So over 1, 5, 10 minutes, and as you can see, the longer, in this case, the longer 11 base pair fragment is here, and that grows bigger, and the shorter one is relatively smaller over the course of the reaction. So basically, we do that both in ordinary water and in deuterated water and ask if there's any difference. And there wasn't. So this shows, I can't see because of things moving around. Okay, can't actually see, I can't remember what is. Okay, so essentially, when we did the experiment in water, we got exactly the same profile as doing the experiment in 100% D2O. There was no difference. So in this experiment, we haven't been able to detect the kinetic isotope effect. So whatever, when we're making DNA in this particular experiment, it doesn't seem to care whether it's hydrogen or deuterium there. It incorporates the base just as well, whether it's hydrogen or deuterium, and doesn't make any more mistakes, if you like, when deuterium is present. So that was a bit of a disappointment. Okay, because it's gone. Okay, so that was a bit of a disappointment. But as I pointed out before, DNA replication is a complicated business, and there's lots of things maybe going on that maybe complicate our interpretation of the experiment. But at the moment, it's our conclusion is there's strong theoretical evidence for proton tunneling in DNA, but does it cause mutation? Well, in our hands, we haven't got any evidence for that yet. It still seems to be that in the more refined in vitro experiment, we don't get any evidence of a kinetic isotope effect, despite Louis' calculations suggesting that we should. So that's it really for science and what I'll be talking about. This is our current quantum biology doctor training group, or a number of them at the Queb's 2022 meeting in Crete. Who's there at the back? There he is there, but Antonio wasn't able to attend the meeting. And that's it. Thank you very much.
What are your questions, folks? I mean, you should talk. Thank you, Johnjoe, for this super cool talk. Folks, what are the
Please.
Are there questions from students? Graham, I know you want to ask questions to Johnjoe, but are there questions from students first? We were going to get to you, Graham. Silence. Okay, Graham, go ahead. Nice to see you.
Yeah, so I've been
Graham.
interested. Good to see you again, Johnjoe. I've been interested in this for a long time, and I just want to see if you've thought of the same kind of things. So there was a big science paper from Tomasetti and Vogelstein a while ago that says cancer is just mostly bad luck.
Yeah.
Do you
created a lot of controversy? Yes, and it's certainly consistent with a random mutagenesis kind of mechanism.
But do you think if your and Jim's original theory about this superposition lasting long enough. Is that viable in a eukaryotic cell with those kinds of turnover numbers, or not?
Well, I'm not in Louis' simulations, and you have to get Louis back to UCLA seminars to get the details of it. But actually, it isn't, because it's a more subtle effect to do with the because it's swapping over. If you, that's not really what's happening. It's present in a superposition of both forms, and the presence of deuterium in the experiment actually changes the proportions because it slows the transition, but it slows them asymmetrically between the two forms. And that's what surprised me. I thought it would be symmetrical, that, okay, if you add to, this is what my own criticism of the experiment, I thought it's not like to work, because if you add deuterium, it's going to slow the transition from the normal form to the tautomate form, but it also slow the backward transition. And therefore you wouldn't get the effect. The equilibrium between the two wouldn't change. But in Louis' simulations, it seemed to be, and I don't really understand particularly well, not really at all, but in his calculations, it seems to be that the deuterium acts asymmetrically. It's on one or the other transitions. So it does change the equilibrium between them. So it doesn't really then depend on the coherence rate, because that's happening all the time. And it's just that the equilibrium concentration or relative equilibrium concentration of them will change with deuterium according to his model.
Okay. Thanks.
Questions from trainees in the call. What are you curious about? You have the man himself who wrote the book that we're following throughout this seminar. Do you have questions for Johnjoe? Maybe even related to the writing of the book or something. Yeah, Johnjoe, they're too
Stunned.
shy.
Stunned. Okay, well, there's a question on the text line. What is the next step in experimentation to figure out if tunneling is related to? Yeah, it's been troubling us. I think it's we have to probe more into different experimental systems, I think, and it could be that the tunneling may be taking place and in our system and may be involved in the mutation, but maybe other things are going on in the mismatch repair system that may be no, that not in that in vitro system actually. I don't know, to be honest. It's one of the experimental systems that we're looking at is that we're thinking of looking at is actually looking at the DNA polymerase and seeing what if we can probe it into the DNA polymerase in a DNA replication reaction and see if we can see what's going on there. But yeah, I'm a little bit stumped for what's the next thing to do, but I'm not going to give up just yet on the idea of tunneling being involved. Or it could be that Louis' calculations are wrong. I mean, maybe tunneling is involved, but it's symmetrical after all. I don't know. That's another possibility. Yeah.
Can you hear me?
I didn't hear any question, but I just heard you say, can you hear me?
Okay, great. My name's Dr. Kieran Johnson. I'm in Wellington, New Zealand.
Oh,
The question I have is deuterium competent to tunnel, or is it too big?
It's too massive. It isn't that it doesn't tunnel, but it tunnels at a low rate. The quantum physicists here will know more about it, but apparently it's because in the tunneling reaction, the mass goes into the exponential, and that really poops on the rate of transition between the two tunneling states, two possible states.
So you could say that the only competent element for tunneling is H1?
Yeah, that's certainly would tunnel. a lot faster, but it tunnels at a very fast. It tunnels through. It has a frequency, and that's very fast. And even deuterium will have a frequency, but it'll be a lot slower. And I guess the question is that whether it makes a difference if it's tunneling so fast, then is it going to make a difference to when a DNA base comes in to be replicated? And that, so far, we're not finding that it does. But yeah,
what I've heard other people say is that the process of glycolysis, Krebs cycle, electron transport chain is selecting for quantum competence of hydrogen or the proton rather than deuterium. And in that process, if you're having a deuterium D2O background, would you be, in a sense, selecting for, due to the other biochemical processes within the organism, the protonation is kind of stopping it from actually doing the tunneling events in the isomeric form or tautom form?
Yeah, I think that relates to something else that's a little bit of puzzle in the system anyway. What is the effect of deuteration on living cells? If we put E. coli directly into 100% D2O, it dies. It doesn't grow.
Which really suggests that without protons, they're in trouble.
Cells care about it, but then there may be so many different things that may be going on. I mean, hydrogen bonds are involved in protein structure. They're constantly having to be broken and reformed as proteins form. So what the effect of deuterium is on that, I'm not sure, but I think it's an interesting question. It's not something that anyone has really nailed. What is the toxicity of deuterium? Why is it that if it's meant to be chemically, they're the same pretty much. So why is deuterium toxic? I don't think we know. Antonio did do a rather cool experiment that we haven't managed to unravel because of COVID. He started the experiment and then COVID hit, where we grew E. coli in increasing levels of deuterium. So you can acclimatize E. coli to high levels of deuterium. It will go up to about 95% deuterium. Grows a bit slower, but it will acclimatize. So it would be interesting to find out what's going on in that acclimatization to deuterium. And we did the experiment, but we haven't been able to look at results because they got locked away in the freezer somewhere under COVID. But I think that would be interesting to find out what is the toxicity of deuterium due to.
Ion channel gets messed up with deuterium, right? Ion channel functioning.
Yeah, that's something we're looking at as well. It should be that deuterium will make a difference because it's actually we're not sure. I think theoretically it has been proposed to make a difference, but the nucleus, you kind of think of the nucleus as being, it's bigger, but it's still tiny. It's still a tiny, tiny, tiny thing. And the difference between a hydrogen nucleus in size and the deuterium nucleus in size is infinitesimal compared to the size of the atom. Because it's, of course, the nucleus is a tiny, tiny fraction of the size of the atom. So size doesn't matter in that case. So why it's proposed, one of our other students is looking at ion passage through ion channels, and to see whether there is a kinetic isotope effect. So we are exploring that. It certainly still goes through. Deuterated water goes through ion channels, but it may go through more slowly. But it's not caused by the size of the nucleus.
Yeah.
That's trivial.
I've got a slightly different proton model. I've gone back into the electromagnetic force within the structure of the proton in order to try to understand tunneling from the various s orbital structure layers and trying to comprehend either the Lyman, Balmer, or Paschen transitions as a way of understanding the wavelength differences between the various points to allow tunneling to occur. And it looks, for me at least, from the calculations that I've done, that the Passion infrared wavelengths around 820 nanometers provide a longer path length for the photon to jump from one place to another. And rather than having the idea of a proton with 3 quarks in the nucleus, I've gone into a mirrored symmetry model within that proton where I've got a positron electron pairing at right angles to each other in order to understand a functional cross-space geometry within the proton structure.
One of the ways that we've been again, Antonio did experiments in another lab where we've been looking at terahertz radiation, because if the frequency of the tunneling is well, it'll have a certain frequency both in ordinary water, and it'll have a different frequency in deuterated water. Can we probe that with terahertz radiation and push it more into the normal or the tautomeric form? And that's another way that we were thinking of probing this system as well. But again, it's been interrupted. Plans to do those kind of experiments. But I think, yeah, looking at the frequencies of transition is an interesting way forward.
I think Graham had his hand up.
Oh,
Yeah, I was just thinking, the way that Judith really beat down on tunneling was wide temperature ranges.
Okay.
Because the tunneling is very temperature independent. Could you go with like extremophiles?
Yeah, we have
all the way
thought
down
about
to cycrophiles.
Yeah, we have thought about doing that, doing experiments with really cool down organisms. Of course, you can't go below freezing point, and you can go up to 113 or so. That's about as far as you can go. And yeah, that's another thing that we thought we might look into. But then you've got to use different polymerases for each of them. So it might be different to stitch the entire experiment together. The other thing that we're trying to do is, and I don't know if anyone has experience with this, one of our students is doing neutron scattering of DNA to see if we can map where the protons are. It will be difficult because whatever state we do, I guess the nucleotides are going to be mostly in the canonical form with a small proportion in the tautomeric form. But with neutron scattering, we can actually map protons. So rather than X-ray crystallography, we're just looking at the electrons essentially. With neutron scattering, we can map the protons, and we have it to have a student working on that, Nermin, at QPTTC.
How much time do we have? There are some questions in the chat. All of these.
Johnjoe, keep going. I think Natsumi is saying that there are some questions on the chat.
Oh, right. Okay. Yeah. Okay. Lots of them.
You can read them from here.
Okay, I can read them out first. Okay. It's been proposed that glycolysis selects for quantum competent H and not deuterium. How would you work in your model of D2O background? Funnily enough, in my non-quantum mechanical work, I do a lot of work with heavy isotopes in metabolism, and certainly it makes a difference if you use heavy isotopes. The incorporation of heavy isotopes compared to light isotopes is different for different metabolic reactions. So some reactions are sensitive to heavy isotopes, such as deuterium versus hydrogen, or C13 versus C12, but not by much. It's normally quite a subtle change of about 5% or so in the products. So I don't think that would be enough in glycolysis. As far as I know, there's no extremely sensitive. It's how they do, how they detect photosynthesis in fossils, because carbon-13, the fixation of carbon has a preference for C12 rather than C13. So in biological carbon, it's got a lower level of C13 than in organic carbon, but it's small. only a percent or so. Okay, thanks. Quantum entanglement, like diseases that come together, for example, Alzheimer's also bring depression and possibly susceptible diabetes. I'm not sure where in chromosomal genes are positioned above these diseases I mentioned, but could be possible that gene entanglement is happening there. I have no idea actually. Alzheimer's disease. We did I think there may be possibilities that Alzheimer's disease involves something quantum mechanical because it's a protein that folds into the wrong structure. And protein folding is certainly a quantum mechanical phenomenon. And I think that does deserve some serious quantum mechanical work done on exactly what's going on there. But I don't think it's been done.
But nothing that you talked about today has to do with entanglement, right?
No. Oh, nothing to do with entanglement. No, nothing to do with entanglement. I don't think anyone has shown entanglement to play a role in biology. I know Vlad Konfetual has managed to entangle an E. coli cell, but that's which is a fantastically technical, wonderfully thing to do, but completely irrelevant to the E. coli cell. It didn't know it was being entangled. As far as I know, no one's demonstrated entanglement playing a role in any biological system. What's the next step? Yeah.
One last question here. Can you tell us about opportunities for undergraduates at the Quantum Biology Doctoral Training Center to do research in the field?
Yeah, we're going to be have four PhD studentships available in 2023. We haven't advertised them yet, but they will be available to theoreticians and to experimentalist posts. So there may actually, there's five. There's another one looking at ion channels as well. So, but it comes with a different funding source, and there may be more. But at the moment, we've got five becoming available in 2023. So keep an eye on our website. Hopefully we'll provide some details on there soon. But I know there's certainly one on ion channels and whether quantum mechanics is involved in ion channels. So if anyone is interested in that question, there's an undergraduate, a graduate who's or an undergraduate who's about to become a graduate in ion channels. We do have one studentship available. It's not yet advertised, but it's available.
Okay, so thank you so much. I think we have to kind of close the meeting soon, but I just wanted to make a quick announcement to the people taking this class. So the next lecture would be not next Friday, but it will be next Monday, November 7th, due to speakers' schedule conflict. It's the same time. Yeah. Same time, 11 a.m. Yeah, so we'll see you again soon. It's going to be about chapter 2. And with that, thank you so much, Professor McFadden, for joining us today.
Thank you for listening. Bye.
Look forward to seeing you soon, Johnjoe. Thank you so much.
Yeah, look forward to it. Bye-bye, everyone.